My second guest on the Plant Blindness series is Graeme Errington. Graeme is the curator at Plantbank, which is at Mount Annan, a secondary site of the Royal Botanical Gardens. Graeme's background is as a horticulturist and an arboriculturist, and he is here today to really talk about the seed collection and conservation activities out at Plantbank at Mount Annan. Welcome, Graeme. Thank you so much for joining us.
Nice to be here.
Could you start by telling us a little bit about Plantbank itself? Because it is the premier seed bank in Australia, isn't it?
Of course. Yes, it's a fantastic facility. It was built about 6 years ago, $21 million budget. The interesting thing about the building is that it brings together components of activities that we've always done at Mount Annan since it opened in 1988. So education and plant conservation research and seed collection. And those are located in one facility, and there's public access, which is a core component of the building, is engagement with public. So the actual area that I work in is a three glass walls, and the public come past and tap on the glass and have a look in. But it's all about engaging with people and getting them to understand a bit more than they knew about plants before they came.
Yeah.
And which is an important part of what botanic gardens do, as opposed to parklands. So
yes, which reminds me actually that Plantbank, our Plant Bank, has a partnership or a very close connection with the Millennium Seed Bank over at Kew Gardens.
Yes.
And actually recently, it was a couple of years ago, I was at Kew and there was a lot of Australian exhibitions and activities. So it seems to be quite a vital and connected and quite lively partnership.
Absolutely.
Yeah?
We have about 2,000 of our accessions, individual seed collections, duplicated at the seed bank in Kew, and we've received a lot of training and technical support, and that's an ongoing building relationship, and it's been critical to the work that we've done. And they have developed that partnership across the globe. I think they've got about 80 partners internationally, and I've been involved with some of those partners in other countries. But it's definitely a global effort, and we share the information because there's a lot to try and learn about the material that we store.
And this is sort of an interesting point that I hadn't really thought about before. So if Kew is that kind of central partner with all these 80 or so partners around the world, do they have a much larger storage facility? Is that why they can accept those duplicates there, as opposed to sort of sharing them equally around?
It depends on the capacity of the partner country.
Yeah.
So for us, we have lots of space, we can process the material, we can do a lot of research on that material, and it's really a backup for us. We send a small portion of our collection for them to store, so that probably doesn't take up a lot of space. But some of the other partners have very limited facilities, or maybe just do the collections and send all of the collection to the UK to be processed, to be tested, for the research and for the storage. So yes, it's a large facility, and they have a lot of programs running with that. And yes. So
let's start with your collection process. You yourself,
yep.
but maybe we could start with how many seed specimens there are at Plantbank, and how you go. Do you literally get in your truck and drive around and visit people?
Yes, absolutely.
And do people contact you, or do you have kind of a policy or a plan, a strategy plan of what you want to collect?
There's a few questions there.
Yeah.
So one of the really key things about the seed collections that we have is that they are supported by in the first instance, a voucher that goes to the herbarium. So a specimen from the plant that we're collecting from, which is lodged with the herbarium, which is part of the Botanic Gardens. And that is a permanent link to the identification of that collection. And then the other key component is the data that goes along with that. So when we're doing a collection, we record a lot of information about the habitat, the associated species there, the soil type, as much information as we can get that is useful for trying to understand the types of environmental factors that might be important in germinating those seeds and growing those seeds. Because an important part of the collection that we still do is to grow that material for display in the living collection. So for that reason, we tend not to take collections from other people. We do nearly all of our collections ourselves.
Oh, that's interesting.
So we have a team that goes out into the field, and there can be quite a long lead time, maybe 12 months or more before you go out and do the collection. So you're planning where to go, when to go. There's certain factors that are important in maximising the value of that seed material. So we want it as mature as possible. That will affect the seed storage, how long we can keep it in storage, and it will have an impact on germination as well. So it's important to be there at the right time, but you don't want to be too late. So we were talking earlier about how some seed is held on plants and some seed is dispersed. If it's held on the plant, like some people might be familiar with, say, a Banksia, which has a cone, which holds the seed for a number of years until there's a fire, which releases the seed. So you pretty much go any time and collect that seed. But other things are dispersed as soon as it's mature. The fruit splits and the seed's dispersed. And so if you're not there at the right time,
you miss the opportunity.
You've missed the opportunity. Animals have taken the seed or it's disappeared into the soil. So timing is important.
So I've been out to the Plantbank a couple of times and been inside the secret rooms with the blue light. So there's the cryo storage room, there's, I forgot what it's called, the
thermogradient
table, and then the tissue culture room.
Maybe
the tissue culture room. Maybe could you just tell us about those three elements? Because I really like this idea. Where, you know, I'm hoping to do a project with the Deputy Dean of UNSW Art and Design, because what we really like is the idea of working with Mount Annan and the Seedbank. But what particularly interests me is the idea of the way scientists work and move and perform their activities, because I think that's a lot of that is behind the scenes. Even though you've got those glass walls, it's still a very intriguing and fascinating process, all your procedures. So could you tell us a little bit about that?
And you're right, and that's one of the things that I and a lot of my colleagues have always been very keen to get people to understand what we do, which was a bit hard when you were locked away in the shed up the back of the nursery. But the beautiful thing about Plantbank is that it allows people to come in and there's interpretation and they can see what's going on. But then we also have tours where, you know, they can get some of the specialists like myself to give them behind the scenes tours where we can give them the detail of what we do. So yeah, I think people are always fascinated when they see the range and diversity of what we do. Now, to answer your original question about some of the processes, there's that basic process of material that comes in and we can seed bank it in a traditional sense. So there's 2 steps to that. And there's about 80% of the Australian flora that we can treat this way. So a lot of species that come from what might be termed a dry land community, like a eucalypt woodland, we can bring those seeds in, we can dry them down in a special drying room where the blue light is. And once the seed moisture content is low enough, then we can freeze those seeds. Now that combination of drying and freezing multiplies the inherent longevity that those seeds have. And so we can multiply that out to many hundreds of years. And now, just to dwell on that for a minute, the reason that's important is because One of the things that we're doing when we go out to collect is to maximise the capture of genetic diversity, as opposed to an agricultural crop, which is bred to be very uniform in its behaviour, seed storage behaviour, growth, harvest, longevity, all of those things are very genetically uniform. Our collections are genetically diverse, and consequently, some of those seeds won't live very long, even within a particular cohort. So by maximising it, we've given the best chance for all of that genetic diversity to be alive in 100 or 200 years, because we don't just want certain genes to survive. So that combination of drying and freezing, most of the species that we deal with, we can put through that. There's a whole range of species that come from rainforest communities that don't have those adaptations. Specifically, the adaptations of the drying is really important. They have a different ecological process where a lot of rainforest species tend to germinate immediately and sit as a seedling. So the seed phase is very short for them. So they either don't like being dried, or if we can dry them, they don't like being frozen. No, they don't like being frozen as well. So they have, some of them have a preference. So what we're doing is trying to find alternatives for those.
And that's the tissue culture room.
That's tissue culture and the cryo storage.
Okay.
So there's a couple of aspects to that too, where with the cryo storage, it's possible to just put either a seed or part of a seed, the embryo, into cryo storage if we pre-treat it, put that away, and then we can store it and protect it. Or one of the other things that we're looking at is growing the material in tissue culture and then just taking a bit of the stem, meristematic tissue, and then treating that and putting that into cryo storage. And so that's really the cutting edge of the work. It's globally cutting edge, significant work that's being done on rainforest species. The Rainforest project that we're doing is a multi-million-dollar project looking at hundreds of species from across Australia. It's globally significant, not being done on this scale anywhere else.
Wow.
And has implications for rainforest conservation across the globe.
Wow.
So
that's exciting.
Yeah, absolutely.
And so...
Do you want me to mention about the thermogradient?
Oh yes, I love the thermogradient thing.
So
it's
we were
amazing.
talking before about temperature response.
Yes, yeah.
And so a lot of things don't have a particular temperature response, so we just put them in a germination cabinet at 20 degrees and they pop out. Yeah. That's fine. Some things need that seasonal temperature, and we can set up something that might be 25 during the day and 5 degrees at night. Yeah. And that alternating temperature will germinate them. Other things we don't know very much about. So we have these two thermogradient plates, which are really cool like piece of equipment.
Yeah, they just look like photocopiers, don't they? Like massive.
They are. They're about 1 metre square, and it's a large aluminium plate which has a temperature gradient across it from 5 degrees to 40 degrees. Yeah. And that gradient runs both ways. So what that does is generate a range of those alternating temperatures from 5 to 40, 10, 35. And so we can put seeds right across that gradient and we can pick up the very specific responses rather than trying to run, say, 30 different germination cabinets and not having enough seed to be able to do that. Yeah. And so what has been found with some of the work that we've done and work that some of my colleagues have done at some of the other national seed banks is that there are species that won't germinate at low temperatures and they won't germinate at high temperatures. So they have this very specific envelope, and it goes back to what we were saying before about them being adapted to a certain climate or a certain environment. One of the interesting things that was found with some of the Western Australian species that occur on top of a mountain, that they're adapted to a particular temperature range and with predicted increases in temperature. Yeah.
And just fluctuation as well.
Yes.
So this
they might,
problem.
if they were halfway up the mountain, they might have a chance to move up and adapt. But if they're already at the top of that mountain, then they're limited in their capacity to change. So, yeah. And even some of the plants that we've been working on in Western Sydney, There's the potential that you get increased temperatures over those key spring and autumn periods, which is where they're saying there'll be probably the biggest increases in temperature.
Yeah.
That will limit germination. So what effect? It might take a long time for the effect of that to be seen.
Yeah, that's right. We just don't know.
Yeah.
Which brings us to our next question, which is the issue of plant blindness. So we talked before about how there has been a little bit of pushback against that term, the idea of blindness, and maybe we could talk a little bit about the problem with the language there. But I think for both of us, we both understand that if plant blindness, which is a kind of compelling term that has been around for 30 years, and it has been, written about and published about in peer-reviewed journals, so it has that kind of background to it. So it's a helpful term to use. And if it is, as I said earlier, the inability of humans to appreciate and acknowledge the plants in their immediate environment, how I know that this is a really important issue for you guys out at the Plantbank. So in terms of these problems of, fluctuating temperatures and how that affects the germination of seeds, how is curing plant blindness going to help all of us? But how are you guys out at the Plantbank doing work that might affect the curing of plant blindness?
So when we were talking about this before, I said that's a term which people are familiar with and it draws attention to it. But as other people have said, it's a much deeper thing than that. But if you try and present the full scope and the depth of some of these things to people in the first instance, it's too much to deal with.
It is.
But one of the things that I always try and get people to understand, and I think this can be challenging, particularly for plants, because of the diversity. So there's more than 24,000 species in Australia. There's 6,500 species in New South Wales. Now, a lot of people would perhaps recognise a gum tree if you pointed it out to them, but do they know that there's like 700 species? Everybody's favourite, the wattle tree. There's nearly 1,000 species of wattles in Australia.
Yeah.
So even when people see plant species and recognise them, they don't understand that diversity. And the reason that diversity is important is because that diversity supports diversity. So the animals that people do recognise, because there's less of them, they're easier to remember. If we
they
had,
look you in the eye.
Well, there's a certain quality like that, and that's one of the reasons why insects get overlooked, and that's where a lot of diversity is. But if there were perhaps 200 species of koalas, then people might have a little bit of difficulty understanding that as well.
That's true.
And so one of the things that we're really focused on as a botanic garden is at that first level is getting people to understand that diversity. So if you come to a botanic gardens as opposed to a park, which will have lots of plants in it, generally speaking, the plants that we have on display have labels with them, and they have a little bit of interpretation, tells you where that came from, which goes back to the voucher that we collected when we did the original collection, and that data. So I can tell you exactly where that tree was collected from. And then the education program that we have is building around understanding the importance of plants, not just to us, but to the animals that we all love and know, and why that's important to us. Even in an urban environment, I think even if people see trees, they don't necessarily understand what those trees give them. They might, and it's a really hot day and they're standing in the shade of it.
So it becomes utilitarian again, doesn't it?
But the other things that trees are constantly doing, they're habitat for urban birds and other urban animals, they're filtering pollution from the local environment, they're ameliorating the local extremes of temperature. These things which they just sit in the background and keep doing, which make our life pleasant. That's
livable.
Livable, And I think it goes back to what we were saying before about the potential situation in the future, that these things will become more important
That's
So
right.
people need to have a greater appreciation and understanding of why it's important to conserve them. Yeah.
Thank you so much, Graeme. That's really been really illuminating, and thanks for sharing your time with us today.
Glad to be here, Prue.